With muscular dystrophy, dys means bad or difficult, and troph means nourish. So muscular dystrophy basically refers to the muscle appearing poorly nourished because of degeneration, which leads to muscle weakness. Under a microscope, a biopsy of tissue shows changes in the muscle itself, but not in the nerve or neuromuscular junction. And this distinguishes muscular dystrophy from other problems that cause muscle weakness as a result of nerve damage, like neuropathies. Muscular dystrophy is actually a group of disorders, all of which are caused by genetic mutations. Within that group, dystrophinopathies are the most common, which includes Duchenne muscular dystrophy, or DMD, and Becker muscular dystrophy, both of which result from mutations in the dystrophin gene. In addition to those two, genetic mutations in other genes are responsible for several dozen other muscular dystrophies, some of which code for proteins that form a protein complex with dystrophin protein. These other muscular dystrophies, therefore, end up causing a lot of the same symptoms as the dystrophinopathies. Now, the fact that both Duchenne and Becker muscular dystrophy result from mutations in the same dystrophin gene means that they're allelic disorders. And when a mutation happens in dystrophin that's severe enough to result in no protein at all, for example, a nonsense or a frameshift mutation, the result is Duchenne muscular dystrophy, which ends up being the more severe of the two, with symptoms usually presenting by age 5. On the other hand, mutations that allow for a misshapen protein to form, like missense mutations, lead to Becker muscular dystrophy, which is basically a milder form of Duchenne muscular dystrophy that presents later on, usually between ages 10 and 20. Alright, so the dystrophin gene is a huge gene on the X chromosome that has 79 exons and is over 2 million base pairs in length. By comparison, most genes only have about 10 exons and are 50,000 base pairs in length. More base pairs and more exons means that there's more chances for mistakes during meiosis, which is when the egg or sperm are created. Most of these gene mutations are deletions or duplications of one or more exons, and a small amount are point mutations. Now, males have one X and one Y chromosome, right? And females have two X chromosomes. This means that muscular dystrophy is way more common in boys, because they only have one copy of the dystrophin gene, and if that copy is defective, it's the only one available to muscle cells, whereas girls with a defective dystrophin gene might have another functional one. Since this is linked to the X chromosome, both Duchenne and Becker muscular dystrophy are called X-linked recessive. In females, though, only one X chromosome gets expressed, and the other is inactivated, called X inactivation or lyonization. Now, if this inactivation's random, you'd expect about half of the female cells to have a functional dystrophin gene, and the other half to have a defective dystrophin gene. And these people are typically asymptomatic. Having said that, if more cells end up with the defective dystrophin gene, and less with the functional one, they can end up being manifesting carriers, meaning that they manifest or they show some symptoms. Alright, so the dystrophin protein links intracellular actin with the dystrophin associated protein complex, which is a cluster of cytoplasmic and cell membrane proteins that are anchored to the extracellular matrix around the muscle cell. Making that link between the cytoskeletal actin and the extracellular matrix stabilizes the sarcolemma, or muscle cell membrane, in the same way that a large wooden support beam running along the roof keeps a house sturdy. Without the support of dystrophin in place, the sarcolemma essentially wilts and becomes unstable. Over time, cellular proteins like creatine kinase, or CK, start escaping the damaged cell, and calcium starts to enter the cell, and this ultimately leads to cell death. In the short term, there's muscle regeneration, resulting in muscle fibers of different sizes. But in the long term, the muscles atrophy and are infiltrated by fat and fibrotic tissue, which leaves them really weak. This process is particularly noticeable in the legs, and children with Duchenne muscular dystrophy begin to walk later in childhood, and they have a waddling gait, and they tend to develop calf pseudohypertrophy, where they have visibly enlarged calves, which are large because of fat and fibrotic tissue rather than muscle tissue. Another classic sign of Duchenne muscular dystrophy is Gower's sign, where if a child's lying down flat on their stomach, they'll try to slowly stand up with the help of their arms, because of weak muscles around the hips and the upper leg. legs. Later symptoms include needing a wheelchair because of severe weakness, developing respiratory failure because of a weak diaphragm, scoliosis, and developing dilated cardiomyopathy and arrhythmias, since the dystrophin protein is also expressed in heart muscle. Unfortunately, these complications often lead to a shortened lifespan. For diagnosis, people with Duchenne or Becker muscular dystrophy often have a high creatine kinase level, and the diagnosis can be confirmed by looking for mutations in dystrophin by either DNA tests or Western blot, as well as having a muscle biopsy with staining for dystrophin. Unfortunately, there are no great treatments for Duchenne or Becker muscular dystrophy. Glucocorticoids can sometimes slow degeneration, but can also result in side effects like excessive weight gain. Other treatments like physical therapy and conditioning can improve quality of life, but they don't reverse the underlying process. Given the possibility of genetic inheritance, counseling parents of the person and understanding the risk of having another child with these conditions is important. About 2/3 of the time the person's mother is a carrier, and the other third of the time the disease is sporadic, which means it's caused by a new mutation. If the mother's a carrier, and we look at our future sons, half or 50% will end up having the mutation. And if we look at our future daughters, half or 50% of them will be carriers for the mutation. Okay, so as a quick recap, the protein dystrophin is super important for stabilizing the muscle cell membrane. Mutations in the dystrophin gene that lead to a loss of dystrophin leads to Duchenne muscular dystrophy, whereas mutations in the dystrophin gene that leads to misshapen dystrophin leads to Becker muscular dystrophy. Thanks for watching. You can help support us by donating on Patreon or subscribing to our channel or telling your friends about us on social media. Spinal muscular atrophy, or SMA, is a genetic disorder where nerve cells in the spinal cord die prematurely. And this causes the muscles that would normally be controlled by those nerves to atrophy or wither away, which causes weakness. When the brain wants a muscle to contract, it sends a signal through an upper motor neuron, which takes the impulse from the brain to the spinal cord, and then through a lower motor neuron, which goes from the spinal cord to the neuromuscular junction, which is where the lower motor neuron touches the muscle cell. The lower motor neurons, which cause voluntary contractions of skeletal muscles, are called alpha motor neurons. And these alpha motor neurons are the ones that die in SMA. Their cell bodies are found in the anterior horn, or front part of the spinal cord, and their axons project from the spinal cord all the way to the muscles they innervate. A group of these neurons is called a motor nerve. If a lower motor neuron dies, or if the entire nerve is injured, the motor unit, which includes the neuron and the muscle fibers it innervates, stops working. Depending on how many muscle fibers stop contracting, there can be overall muscle weakness, or in an extreme situation, a flaccid or low-tone paralysis. This denervated muscle also atrophies over time, a classic example of use it or lose it. This contrasts with the increased muscle tone and spasticity that develops after an upper motor neuron's damaged. When a lot of these muscle fibers are affected, fasciculations can happen, which are spontaneous, involuntary muscle contractions. Alpha motor neurons also carry the signal for muscle contraction in deep tendon reflexes, like the knee-jerk reflex, and they diminish or disappear when alpha motor neurons are damaged. Now, it turns out that there are a few types and subtypes of SMA. Type 1A, congenital SMA, is the most severe of all, and it starts even before birth, when mothers might notice decreased fetal movements. SMA type 1B, also called infantile SMA or Wernicke-Hoffmann disease, is the classic form where babies often appear normal at birth, and then in the first few weeks of life develop hypotonia, or low muscle tone. These infants have progressive weakness, which is worse proximally than distally, and is initially more obvious in the legs, making it hard for them to do things like sit up. They can also have weakness in the muscles involved in sucking, chewing, and swallowing, and as a result they can have difficulty taking milk, eating foods, or even safely swallowing their own secretions, which can lead to aspiration. The weakness can also affect the chest wall muscles and diaphragm, leading to breathing difficulty and eventually respiratory failure. For these reasons, most of these babies survive only a few years. SMA types 2, 3, and 4 are each successively milder and have a later age of onset. In addition to muscle weakness, feeding problems, and breathing difficulties, chronic symptoms of SMA include scoliosis due to poor muscle support of the spine, and extremely thin limbs due to muscle wasting. The different types of SMA all result from the same homozygous deletion of the survival motor neuron 1 gene, or SMN1 gene, on chromosome 5, and this is inherited in an autosomal recessive pattern. The SMN protein from the SMN1 gene is expressed in all cells. For one, the SMN protein is a component of the spliceosome, a molecular machine that cuts the introns out of pre-messenger RNAs. SMN also blocks caspases, which are proteins involved in apoptosis, or programmed cell death. So lacking SMN might also enhance apoptosis. SMN protein is particularly important for alpha motor neurons, but the exact mechanism relating the protein to the function of those cells is still unclear. Now, the genetics help explain the continuum of severity that we see across SMA, and it has to do with the SMN2 gene, which is a pseudogene that sits next to SMN1 on chromosome 5. Pseudogenes are mutated copies of genes that arose during evolution, and are less functional or non-functional versions of their counterparts. SMN2 is more than 99% identical to SMN1, but has one important change in exon 7, called C840CT. And this means that while the 840th nucleotide is a C in SMN1, it's a T in SMN2. And this tiny mutation results in exon 7 being spliced out of the majority of SMN2 mRNA. Not having exon 7 means this SMN2 gene churns out SMN proteins that mostly get degraded, with only a couple full-length functional SMN proteins, relative to SMN1, which churns out only functional SMN proteins. Furthermore, often people might have multiple duplications of the SMN genes, which might result in several copies of SMN2, and this SMN2 copy number actually varies quite a bit in a population. Now, all this is important because patients with SMA have no functioning copies of SMN1. And since SMN2 still makes a small amount of functional SMN protein, the number of copies of SMN2 determines the severity of spinal muscular atrophy. More copies means more SMN protein, and a milder SMA phenotype. So, as an example, a patient with 2 SMN2 genes might have infantile SMA, whereas a patient with 4 SMN2 genes might have a milder subtype. Treatment for SMA has historically been supportive, like giving infants nutrition through a feeding tube, as well as respiratory support to help with muscle stiffness and strengthen respiratory muscles. A relatively new therapy for SMA is called nusinersen. Nusinersen is an antisense oligonucleotide that binds to the SMN2 pre-mRNA and prevents exon 7 from being removed, which allows the SMN2 mRNA to get expressed, ultimately making a more normal amount of SMN protein. Alright, as a quick recap. Spinal muscular atrophy is an autosomal recessive disorder where a deletion of the SMN1 gene causes alpha motor neurons in the spinal cord to die, resulting in muscle weakness and atrophy, and in more severe cases, respiratory failure and death.